Hello all! Today we'll talk about the second qualitative reaction from Biochemistry's Biochemistry box. We'll show you how this reaction detects the presence of proteins in food and various solutions. We have a box that contains 3 experiments and we will use reagents needed for the second experiment. We took a box without included food ingredients, as we can find egg and butter in our own fridge. So, let's take a look inside the Biochemistry's box, which contains beakers, gloves, spoons and pipettes, and reagents, sodium hydroxide and copper sulfate. Proteins are the main macromolecules in our body, both structural and functional. They consist of long chains of amino acids connected by so-called peptide bonds. 8 out of 20 different amino acids found in proteins are indispensable amino acids, which means that human organisms are not able to make them from scratch. And that's why proteins are essential nutrients for us. Different sequences of amino acids in proteins lead to different functions, and proteins play a wide range of roles within organisms. If you remember, in our previous video, we used an enzyme amylase from our saliva to deconstruct the starch in the beaker. Proteins can be detected in food and solutions by a qualitative reaction. This reaction requires copper sulfate and sodium hydroxide, because copper ions interact with peptide bond of protein and form a violet-colored complex. This complex appears only in alkaline conditions, in our case, in the presence of sodium hydroxide. Gloves are obligatory here, as we are going to work with sodium hydroxide. So, like in the previous experiment, let's take 3 beakers with melted butter, egg white and starch and perform our experiments. Next, using a pipette, add copper sulfate into each beaker. And then mix it. We start to see active sedimentation only in the middle one, with the egg white, which contains a lot of proteins. Such a reaction indicates that proteins were denaturated in the presence of heavy metal salts, copper sulfate in our case. Denaturation disrupts the bonds necessary for the normal structure of the protein and uncoils it into a random shape. While cooking, we are used to experiencing temperature denaturation, when eggs or meat change structure and color. Here, we observe the same process induced chemically by heavy metal salts. Now, carefully add several drops of sodium hydroxide into the beakers. Mix it intensively. After intensive mixing, we see that the color in the beaker with denaturated egg white has become intensively violet. That means that egg white contains a lot of proteins, especially in comparison with a completely protein-free starch solution or low-protein butter. Thus, using such an approach, it is possible to indicate the presence of any protein, because peptide bonds are a universal feature of all proteins. You can try it with some protein-rich liquid food, bone broth, protein shakes or yogurt. Thank you for being with us. See you on the third biochemistry lesson from Biobranches.